the bottom in sight. This is dive number 4.
A lot of little fish down there. What's going on?
Of the Windows to the Deep 2018 expedition, we are aboard the Okeanos Explorer. Well off the coast of, we're actually off the coast of Georgia at this point.
You can hold out 15 or 20.
Slowing down. I'm gonna bring my camera up, get you into better view.
Well, that's a great sign. We sat down on a rock.
Didn't have to search.
It looks like we have a cutthroat eel there, Synaphobranchid, just in front of the rock.
We're already seeing more fish than we've seen in the last 2 dives.
Yeah.
And Sirius is stopped with a delta of 12.
Say again?
Sirius is stopped with a delta of 12.
Great. I'll take a couple more meters here to get to the bottom.
Video, which one was that you said I was on?
One last look at the chip chart for Roland.
It doesn't look like that. On the only thing that I have on is OKEX. Huh. It doesn't look like that. I don't know what to do about it.
Tilting up for you. How's that? Okay. Okay. All right, co-pilot, can you stow the arm for us?
Yep.
How does that sound? Thank you, Roland.
So today we're going to be waiting on the scientists to collect the water, correct? We're not doing like a test run right now?
I don't think so, yeah.
Okay.
It's true.
It'll be there tonight, though. It'll still be there.
What's
this
that?
Afternoon.
What's that?
That layer.
So before you get too stowed, you want to get outboard.
Yep.
Because if you try to swing around and you're too stowed, you can like run the fingers along the
Good morning, everyone.
Like that.
And then you can use
Hope
this
that this is
over here to
your 4th visit
boxes.
to our site. We are on our 4th dive, and we're excited already to see some rocks.
Drawer in, boxes out, all that
pretty
stuff.
much in search of rocky habitat, because that's where the megafauna that we like to find.
So we're done with this camera,
that's
so
where they like to live. So we are off the Georgia coast, about, what, 200 kilometers maybe?
Get started here.
I should have checked the distance. I'm not exactly sure.
We're heading off. We can get a couple of zooms.
We're checking now. And we are at a feature called the Blake Escarpment. We're 179 nautical miles.
Okay, watch lead, pilot. Sorry for the little hold up this morning. Because of that thermocline and everything, our camera temperatures didn't come down to a level that video was ready to white balance. So we wanted to do one last double check on bottom, and we're all set now. So anything you see here, we're ready to start zooming.
Excellent. So we're ready to roll.
That's affirmative.
It looks like we have a sea cucumber on the left side of the screen. Synaphobranchid eel. By the way, this is Cheryl Morrison from the U.S. Geological Survey.
Yeah, we're seeing so much right away that we haven't even had time to introduce ourselves. And this is Leslie Sauter of the College of Charleston. I'm the geology science lead.
Seems like they want to look at this.
And we're at a depth of 1,321
You want to have a peek at this video?
meters. the Blake Escarpment South. So this is just further south than we were on our first dive, and same or similar feature.
Similar feature, slightly shallower. And we're kicking off the dive with a wonderful view of a fun creature.
The sea cucumber, Holothurian. And they basically comb the sediments for food, and they'll eat some sediments, and you can see it passing through their gut sometimes with their translucent bodies.
Yeah, we're ready to go. I'm ready to go.
Let's do it.
Video's clear.
20 meters in.
Okay, I was getting stable.
And if you've never
so you
seen one, you'd probably be surprised to find that they are relatives of sea stars and urchins and brittle stars and crinoids. They're in the phylum Echinodermata.
I want to see his other side.
Bridge, RV Nav.
Looks like he might be trying to swim away.
Go ahead.
Hey, we'd like to put in a ship move.
Okay.
Okay, we're looking at
If
2
you've been
0
watching
meters
Okeanos
at
videos over the
275
years, you've
degrees,
probably
0
seen their
2 knots.
beautiful swimming.
Okay, 20 meters at 275, speed decimal 2.
Good copy.
We saw one yesterday, actually, didn't we?
We saw some that were on the seafloor like this, and then one that was swimming, and it's quite graceful and
You see
beautiful.
what looks like fishing line next to that black loop
He turned
in the
around
upper
for
right?
us.
No, I don't see any fishing line. I'm looking for it now.
You're looking for fishing line?
Would you like me to set your lights for flying?
See swirls up there at the top corner.
And the
Oh,
red
yeah, I
dots
think
you
that's
see on the
just-
screen, you'll see them periodically. We turn them on and off throughout the dive. There are 2 laser beams that we use as our measuring tool. They're 10 centimeters apart. There's a nice view. Glad he turned around for us. It's like the
Oh, he's evacuating.
current is kind of pushing him along.
ROV nav bridge, move initiated.
Copy that.
And you can see what he has
That's-
left behind.
This is evacuations.
Little package in the sand, illustrating, I think that's what he's actually doing right now.
Oh, yeah.
Here's some great behavior captured live. He did not put the do not disturb sign on the door.
It's quite impressive, actually.
It is quite impressive. Oh my. There we go.
Wow, here he goes.
There he goes.
He's much lighter now.
Yeah.
Yes. That must have followed his cup of coffee in the morning.
Okay, we'll let him go. He seems irritable. This morning?
Oh, look at that yellow streamer right there.
Yes.
Center screen.
Yeah.
I don't know
sure
if
we
we've
can.
ever found the deposit that he just made. I'm trying to find a term that I can use online. But
Defecation.
his defecation, yeah, I was going to say.
Just getting little
often-
bounces from Sirius, sorry.
We do, not often, but periodically
down.
in the rock record or, deep within sediments when we have dug away
Something up.
through them, we find
I try to get a little
remains
closer and I'll
of
just bounce against
feces,
the
fecal matter.
tether here.
And if they get preserved in the
Just
rock
gonna
record
drop down.
themselves,
Do
meaning
you want
they've
me to come
turned
down a
into
little bit
stone
so you have a
themselves,
little
we
bit
call
more,
them
or-
coprolites.
You got it, yeah.
Yeah, I can come down.
I don't believe
Can I zoom you into 30
we've
meters
seen
there,
a
pilot? I'm
coprolite
sorry.
of a
Sitting on top of a
holothurian.
cool
It's
feature.
unlikely that that would be preserved.
There you go, video. Sorry about the bouncing around.
Okay, let's look at the lower section, larger polyps.
Okay.
There's still
Yeah,
Look
this
like
is.
the...
I haven't
near the
seen
base
anything this
there, or-
color before.
Yeah.
Oh, okay.
Is that a black coral, maybe?
Okay. Come down to a Delta 12, so you have a little bit more room.
Thanks.
Yeah, I don't think we've seen this.
No,
Yeah.
definitely not. I don't know if I've ever seen anything quite that color down here.
Yeah, hey guys, this is Peter from NOAA. That does look like an unbranched black coral. I've seen some of this yellowish green coloration in these before. The unbranched ones are
You want me to tack on 10 more
usually
meters to the end of that one, so that you had a total of 30?
Great, thank you,
Sure.
Peter. That was what I was thinking. But
Bridge, RV Nav.
I've seen them
Go
more
ahead, RV now.
orange.
Hey, can we add an
usually,
extra
or
10 meters
white,
to the end of that
right?
same direction, 275?
Yep.
Adding 10 meters.
What I was
Thank
familiar
you.
with too.
Yeah,
A
interesting.
lot of these corals seem to have some kind of plasticity in terms of their coloration.
Right.
Okay.
Yeah, in the Gulf of Mexico,
ROV
we
net
see
bridge, 10 meters has been added.
orange,
Thank you.
black corals in the Liopathes genus, and then white individuals and kind of all of the shades in between, salmon colors, and at first we were wondering whether there was a
little worm
different
tube
genetics
immediately
behind
to the
each
right
of
of it.
those, but after a lot of sampling and genetics work, it doesn't seem like there's a genetic basis to the color difference.
Well, maybe it's a crinoid.
I don't pretend to know these things. Just kidding.
So we do have our first coral of the day. That
Yes,
didn't take long.
it's very exciting.
Yeah.
Good here, Roland?
Yep, clear.
right.
Science is requesting a close-up of the fecal matter.
I'll take credit for that. Poor Cheryl's looking at me.
Whatever floats your boat.
Is this in here, I
I'm
think?
sorry.
Coming a little closer.
Straight ahead.
Yeah, I think that's it, right next to the lasers.
And the
right?
reason
oh, no,
I
see it
ask
up there.
is in the
Fish
fossil
in there, too.
record, if this is sort of a characteristic pattern of how
Gotta
holothurians
get right on top of
defecate,
it.
it potentially could be fossilized. And if we found that in the
There's another one to
record,
the left
then we would know
immediately
it
as
was from a
well.
holothurian. We may not know what created it. That type of
Come on in.
fossil is not of the actual living organism, of course, but it is a trace fossil. And as I said earlier, if it is fossilized, we call it a coprolite. And because that's quite characteristic if that were to become fossilized somehow. Maybe a mud layer deposited over it, and then it got buried through time. It might well become
Gonna
preserved.
rotate left and get the second one.
So Leslie, how
Just another
long
one,
do you
you said?
think that
Yep.
structure would stay in place versus kind
Plus
of getting
direction.
weathered out or
It's on the
blown
edge. Oh, yeah.
apart by the
I see it.
currents?
Well, it seems heavy, so I
There's another one.
And there's a crab above diagonally from that.
This we found their bathroom.
we might.
Holothurian restroom.
That's pretty cool.
Is it from the same holothurian?
Yep.
Is he
Oh,
big?
there's several
Yeah.
of the Stichopathes too. 4 of them on the top left.
So maybe the Stichopathes are somehow feeding off of the organic matter that comes. That's a far shot, I know.
So this one looks like it was a little bit older and is starting
Might
to
have broken
fall
down
apart.
a little bit, yeah.
Okay, you want to go up to the Stichopathes?
Certainly,
And the crawling crab?
in an environment like this, if it's exposed to the seawater for a long time, then bacteria would begin to degrade the
crab?
Straight
organics
above?
in
Right next to the black
the fecal matter itself.
material.
And we did notice on
Oh, I see
the
it.
descent that this area is quite well oxygenated. In a low oxygen environment, that would have a good chance of being preserved because it wouldn't
I
have
have you
been
there at 61
degraded by
degrees, coming
bacteria.
up a little bit to keep the tether
Great.
in a good position.
Oh, is this a pycnogonid or not?
Well, there's a crab there.
That's just a crab, okay.
Yeah. It looks like a decorator crab that's got a lot of things he's holding on to blend in with the background, camouflage.
Oh, yeah. It's just
Someone has to tell him part of camouflaging is not moving.
what is the larger organism on his back?
That might, is it a sponge?
Got you there at 7
Thanks.
degrees.
Got a big rock behind me, too. Thought he was going to run into the view.
Oh, okay.
Just be careful as you come backwards.
Yep.
I think video's clear.
Feature directly
Yeah, there
behind
are a
you.
lot
actually
of
gonna show that to
species that will blend in with their environment in hopes
All right,
of
before we
not
take
becoming
off, I
somebody's
just wanna
lunch.
take a look behind us here.
There are shrimps on gorgonian corals, other crustaceans that will take on the same exact color
Should be able
of
to
the
get
coral,
pretty
and
close.
hopefully they'll blend in and not wind up on the radar of predators that are coming by. And then the decorator crab is
Go ahead,
one
Watchley.
that will just take organisms or pieces of things that he finds on the seafloor
Yeah, okay.
and attach them to his back
I'm just
on these
spinning
little
around here.
little,
We've got a little outcrop.
kind of
looking. It actually
Velcro-y
looks bigger at first, but
protrusions, and then
There's
so
a
he
good
can just
sized
hide.
Right there at
clump
90.
here.
And you know, if he
Something to
moves
look
to a
at.
different place, he can change out what he has on his back and make sure he's blending in.
And then you wanted to, for possible collection, you want to
Oh, look,
investigate
we
the other
have
smaller rocks?
This is the Storopathes, I guess, again. Some black corals.
Yeah.
Oh, and there's a
Yep.
stalked crinoid in the bottom of the rock, and we have a Sideris pencil urchin maybe on the lower right, and a yellow sponge on the right side. But I believe that all of those orange corals that might be the Storopathes that we saw on the first dive.
This is beautiful.
And is Peter still on the line?
Yes, I'm still here.
Would a collection of one of these black corals be a good idea? Or at least a
I think
piece?
it'd be a great idea, especially because there's several of them. I hate to take the only one, you know.
Right.
But
Would
with 4 or 5, if you can set up and get a close-up image with lasers,
Coming
that
a
one
little
in the
closer on the
center
back
of
of
the
the
frame
there, I guess.
right
There's
now.
also a stalked crinoid on the right.
Yeah, I see that.
There's a lot of stuff here.
You want to come in a little on this guy?
Pilot, this is watch 1.
No,
Go
the
ahead.
ship is stopped, correct?
Would it be possible to try to set up and collect one of these black corals look like feathers that we're looking at now?
This right here in front of us?
Yes.
Okay. Yeah, it would be possible.
Might want to also image that one to the left. They may change their mind.
Yep.
Yeah, actually, Leslie is commenting that the ones that are to the left of this might actually be better because we might be able to knock off a rock as well and get a geology sample as well as the coral sample. Looks like there's a scale worm at the base of this.
Yeah, and this is Peter from
Going
NOAA
full screen.
again. If we could collect an entire colony, you know, as close to the base or including the rock.
Yes, that's what we're going to see if we can knock off.
I have you there at
We want to
95,
have
but the ship is
the
not
whole
moving.
thing.
Will you want me to
Yes,
spin around and
definitely.
get you in a better view, or are we fine?
And Scott Francis commented that there might be Storopathes as well as Bathypathes on this rock. So we might have
Okay,
two
video's clear.
genera of black coral right
Great.
here. So
We'll go
it's a
over
good
for the
idea to
Storopathes here.
take a look
Several
around
crinoids in
and
the shot.
kind of document that there is more than one species. Oh yeah, so
Yeah, if we could try to knock that rock and see if we could get the entire.
Gonna go ahead and take
We
the
might even
lasers
get
out.
2 different corals.
Lasers are clear.
Now, are these both
Lasers out.
black corals? Did you say?
they're both black corals, but I
Okay.
think they're in different genera.
Yeah, they're
I don't
definitely
think that rock
different.
will
aren't
fit
they?
in the
So,
Biobox.
Scott, am I correct that the one on the right that we're looking at now
broken the
is
bad
the
news yet.
Storopathes, and the one that we were just looking
break
at
it, man.
is Bathypathes?
No.
Yep, Storopathes is this one here, and the Bathypathes is the one on the left.
Do you want a little more light, Roland?
Yes, let's try that.
You
Also,
want to turn on the
Yeah, you
fog
can
lights
see
for
how
us?
the one has a pinnate branching. All the branches come off the main stem.
Just
a
That's the
two
one
shot?
on the left.
Right.
Sorry.
One of
Video,
the
you ready for
characteristics
the,
of Bathypathes.
Yes,
Fog
go for
lights?
go for
Whereas
lights.
this one
Coming on
has, kind of secondary branching.
Lights are on.
That must be a characteristic of store.
Slight tilt up.
Tilting
Two-shot.
up.
Something there behind it as well.
Yeah.
And if we could sort of look between the two so we can show that those corals behind.
Sure.
Oh, and that's another little octocoral coral down there between them. Is that a stoloniferous octocoral, I imagine?
And a nice pteropod shell in the foreground. Actually, it appears to be broken. Yeah, this would be a great sample if we...
Okay.
If these rocks are at all like what we found on the Blake Escarpment North dive. They are mudstones encrusted with this very dark...
Is there anything else we want to look at here?
No, I think we're good.
Iron manganese crust. Clear. And they were really quite easy to break. In fact, some of them fell apart on deck. So it'll be interesting to see if this type of rock is similar.
Yep. Yeah.
Lasers coming on.
So we'll know when we hit it. It'll break very easily if
Lasers
it's similar and if it's very resistant to breaking.
You want to just frame it with the lasers for a second?
Something different.
Roland.
Might be more of this manganese oxide.
Yeah, that's good.
Manganese iron oxide as a full layer, as a pavement, instead of just encrusting some other rock.
Okay, I'm set when you are.
Right. We happy with the fog lights?
Yeah.
Coming off video? Fog lights coming off? No?
Notice there's several red shrimp hanging around here as well. Gosh, this one rock is a little oasis.
Yeah, they always seem to be.
Yeah.
But this one even more so than what we've seen to date.
There are also several different sponge species on the rock. See a circular round one, kind of similar to what we were seeing yesterday.
Looked like the entire section moved.
Yeah, so I'm just going to try to get a gauge for size on that. There's no way it's going to fit in a bio box.
No.
So, unless this is really soft stuff, which if it's manganese crust, I don't imagine it will be, but I can try just squishing this side a little bit and see if it breaks off.
Yeah. Just kind of stepping on it.
we don't like using the manipulator to break rocks just because it can damage the jaw cylinder. It can actually bend the jaw cylinder, cause a major leak, and we'll not be able to dive anymore, so we won't want to do that.
Definitely broke.
Did it?
I think it just moved.
I think it just moved.
I think this is not what we found the other day, because any kind of...
So, unfortunately, that's
so we're gonna need
too large for what we have in terms of collection storage, and there's no way for us to break it. So, if there's a specific coral you'd like us to pluck off of here, we can. I also have, as long as I didn't land on it too much, might be below me, like you can see it. This is that's a big rock too, never mind. Caught a glimpse of it, thought it might be smaller.
Peter, would there be a preference to collect one of these species over the other, or should we try for a piece of each species?
You know, I am not a black coral expert, but my impression is that the Bassopathes is fairly common, and the Stauropathes is not very well documented. Maybe Scott France could weigh in.
Yeah, he said that he's not very familiar with the collection of these species in this area. They've collected them on the New England seamounts. They weren't allowed to collect them on the Bahamas escarpment because of they lacked CITES permits.
Interesting. Well, in that case, I would say this is a golden opportunity to collect one of each.
One of each, that'd be great.
Yeah, we'll probably be fairly efficient, since if we set up to collect one, we can collect both. And again, we don't have to take the whole coral. We can just collect a few branches or a little piece of it, and then the coral will remain and continue to grow, and we'll have a lot of information from it.
Want to spin up the hydraulics?
They are small colonies, so it might be hard to get.
Actually, can you confirm we're going to make a collection here?
That's true.
I do want to reiterate that if it is a new species, it's ideal to have an entire colony. And because we have a situation here with several, we would know that they would continue.
Pilot, we're going to take a snip of each of those 2 corals, please.
So, okay. So I'm assuming the one just right of the lasers, the little bit darker, older Bathypathes, and then the yellowish one on the left here.
Yeah, we're going to take a snip of each of the 2 next to each other.
Okay.
On the left.
So you prefer those 2 over the one on the right?
Sounds good.
Copy that. Let me just turn my heading a smidge.
Oh. There's a small fish behind the rock.
I thought it was a tripod. He was so still, but I don't think that now because now he's moving.
Okay.
And a sea star on the right, beyond.
Arms live.
Would the forward lights help with the shadow?
No, we don't need those lights, sorry.
Copy that.
I think it's too low. So I wasn't going to try to grab both at once. That doesn't seem to make sense to me unless that's what you're hoping for. Okay. Is that a good section of this one? Okay.
We're looking to go into the port bio box.
Yeah, we can go port outer.
Okay. Ready to extend the drawer when you are?
Yeah, go ahead.
Coming out.
So despite having come through quite a turbid portion of the water column, just shy of 800 meters, the visibility is beautiful down here. Not very much suspended particles in the water column at all.
And coming back in.
Great. Okay.
Drawer is fully retracted.
So now the pilots are collecting a piece of the bathypathes. We had two different genera of black corals here on this one rocky outcrop. It's definitely a little oasis of life with shrimps. I saw a squat lobster in the back of the rock at the top of our screen now. Many sponge species. We've seen shrimps. I think I might have seen either a brittle starfish or a bracingid.
Same box.
Correct.
Fully extended.
We'll be able to tell them apart at the surface, so.
Retracting the box, the drawer. Oh. It's floating
Pilot,
away.
this is watch one.
What?
It floated away.
What did?
The sample.
Well then, let's close the drawer. So we only got, the other one floated out of the box?
I believe it did.
I don't know. I didn't see.
Do we have any replay on the mini Zeus to see if it floated away, Roland?
This is kind of a notorious thing with black corals. They're very, very light and will get picked up by the currents very easily. So it's hard to actually put them down because they just want to flow away.
That's the first one.
Yeah, the first piece is.
So we're trying to make sure that we actually got that sample of bathypathes into a bio box.
It was on the bottom, and the drawer closed. And then the next one.
Stream 3 has the replay.
Might be better to.
Close. Good. Ooh.
How did that look, Art? Did you see?
It definitely
looks like it did.
It's definitely out. It's floating
yeah,
right there.
There it is.
Yep.
So that's gone. That one floated up and out of the box on closure.
Is there enough left on the one that you have taken from that we could give another shot? Or should we pivot over and take a sample of the bathypathes on the right
Good catch
side?
there, Lars.
Well, it actually wasn't a catch.
I would say there's like a little stem and 2 branches of the other one. And I don't know if I could grab it or not. It's pretty close to the rock. I can try it if you don't want to damage the other coral.
Yeah. Yeah,
you
it
can
might
zoom
help
in a
to
little
zoom
just
and
to
see how
frame
much
it.
is left there.
You can see it's like a stem and 2 branches.
Yeah, I mean, it would be ideal to just take the rest of that if possible, but it also is probably more likely to float away again because it's so tiny. So, yeah, it's a tough choice, but maybe going to the other one would be better.
Okay.
Yeah, this
Sounds
is
like a
Peter.
plan.
That would be my preference.
Sorry, we lost the first one.
To go for the bathypathes on the right.
Yeah, okay. Ooh, and we have... Is that a conger eel on the right that's swimming by?
Have not seen that yet.
It's definitely a lot more stout than a cutthroat eel. And conger eels are quite common as we get more shallow. We'll see a lot of those in the upcoming weeks. Yeah, we saw some stalked crinoids when we were coming around the rock, and they're on the right-hand side of your screen, of your view now.
I don't know how Dan got lucky enough to not have any trouble with this yesterday. No, it's not jumping. It's just not working. It's just not indexing.
Here,
It's.
nobody believed me.
I still don't believe you.
So for anybody just joining us, we sat down, just reached the bottom very close to this big rock outcrop, and it's got lots of different species on it, and so we're taking representative samples of 2 black coral genera that are occurring here, and this is Bathypathes. And black corals are usually not
Very nice.
black in color, but if you were to remove the tissue from the outside, their skeleton is a
we don't want that other one to float away. I don't know. I'd rather put
material.
2 floaty ones in the same
That's
box
how they get their
rather
name.
than not being able to open the box again.
When you're ready, I'll extend the drawer.
Yeah, we'll just move quickly here. I guess we should double check with watch lead that we're making the right decision on that.
Yep.
Watch lead pilot.
Go ahead, pilot.
Our assumption at this point is we're going to try to put this one back in that same box again, assuming that if we open the starboard and put this in the starboard, you won't want to open either if you have 2 floating objects. If we put it here, then you just know not to open the port box for the day.
That sounds like a good
and
plan.
leave your starboard open for non-floating objects.
That sounds great. Thanks,
Okay.
pilot.
Sounds good.
Extending the drawer. Oh.
That's the camera. I don't know what's happening.
Have a nice eel to entertain us.
Yeah.
Wow,
We got to
dude.
switch this back.
Yeah, sorry.
That's all right.
I believe you just did.
Oh, thanks.
Extending the drawer.
Thanks.
Okay, it's full extension.
I think we know where the eel lives.
It's coming up.
Let it settle at the bottom. Or I think it's going down.
Yeah, now it's going down.
All right. Retracting drawer.
Yep. That one's in.
And it looks like we got it. Good
Good eye.
job. Good job.
That would have been strange to
All right.
go back later and
Retracted.
find no coral. Nice work, Lars.
Let's see if we can get a video, if we can get a close-up. A little low.
Thank you, video.
Go ahead, watch lead. Yeah.
Well, we're seeing so
So
much
what
at
we
this
need to do is,
first
for
point
some reason,
of
there's
our
something wrong
dive
with
that
that.
we haven't
Call
covered
us, so
any
what I've
ground.
done is to duplicate it
That's
on
what they
what's
told
called
us earlier. I can ask
side
them if we're
video
ready to move. Actually, this is Nav.
So if you
Hey, were
If
we
you need- if
interested
you
in
can't
taking
find
a
it, I can route it for
Niskin
you.
sample here as well? 3 Niskin valves. We can do that. We will do that right now.
Okay.
That's you, man.
That's a reset fire. Reset fire.
Yeah. Yeah. First one may be already
so what
reset,
you need to
but-
do is to take it to Sci Video Router.
Okay.
It's an alternate that I did
Where
because
is
you
the,
can't get it to come
there's the
up, then I
camera
can
there.
route it
Ready
for
to
you.
fire the first one.
Yep.
Yep.
Firing.
Okay, copy.
Here, hydraulics.
Okay.
Okay, good.
So
Yep.
1,
And then resetting.
There you go.
Firing the second bottle now.
Okay, looks like I need to
Yep,
reboot
fired.
your
And resetting
unit
again.
there.
And firing the third bottle now. Alright. And then we just leave it, next sample, reset fire.
Yep.
Alright. Disabling valve pack and ramping down.
Go ahead,
for you.
watch. 1. Absolutely. All 3 Niskins have captured water, and off we go.
Great. So we just took water
You all
samples
set,
as
Nav?
well here. We
Can you
have
take off?
Niskin
yeah.
bottles on the side of the
gonna
ROV,
hang out a second
and
longer?
they will
Yeah,
close
just
and
give us 1
capture
second.
water that is right from this area where we're looking at the corals.
Okay, you're good.
Great.
And you can actually tell a lot about the environment that the corals live in from.
This guy's big, huh? Oh, it's on already. That's my bad.
Lasers on? Yeah, they are.
I just
Got another
didn't see them at
cutthroat
first,
eel
and accidentally
here.
turned them off. So they got back on. Give them a scan here. Okay, we killed
After the
them.
lasers.
Yep,
Coming
And
off.
this is another cutthroat eel,
Lasers are off.
Snappabranchus.
coming around his head. Oh, maybe. He allows us. You can come in
You
partial
got
video.
that rock feature here, by the
Yeah,
way.
it's short. I'm above it, I think. I hope.
And then Ken Sulak from the U.S. Geological Survey commented that conger eels wouldn't be this deep. They don't generally go below 1,000 meters, so we won't see those today. But definitely later in other dives.
Or if we do, it'll be
A
something
depth record.
never
yeah.
known. A depth record.
Is that a xenophyophora there?
That
In front
looks
of-
like one of the things we saw yesterday, isn't
Yeah.
it? That one of our chat room specialists. Do you remember who sent that to us about the xenophyophora? Or
It
maybe
was
you?
Bob Carney.
Bob Carney.
Yeah.
And what is it again? It's a single-celled organism, right?
Yes.
Can you tell us about it?
I haven't seen that
Bring it
many
up
of them
camera
in
to give you an
my
idea.
times
Great.
looking at the deep sea floor, but they are actually common in some areas, and it's a single cell organism that's part of the foraminifera group, and they create these structures, and the structures actually become habitat for other species as well.
We'll get a view of the tail before we... A caudal fin. Learning my
Let
fin
him swim
names.
through.
Okay.
Oh, and there was a little squat lobster that was in a burrow, the bottom side of your screen.
Okay.
And you can see some echinoderms, maybe some sea urchins that are also scattered. So this is more of what we expected at the very beginning of the dive, just a flat plane of sediments. These are
Yeah,
likely
didn't quite get
muds
beyond
of
that
fine
shelf, did
grain.
we?
It's kind of characteristic of everything we've seen. But as we move along, we're just at the very beginning of our transect
Small fish,
for the
center screen
dive.
up.
We're hoping to encounter
from
some
here?
Yep.
Go ahead.
fairly steep slope that would be the edges of flat-lying rocks of the continental margin.
I view that at
And
55
this is
degrees.
where they
Perfect.
outcrop, meaning that they have exposures. And so they become very suitable habitat for sponges and corals and other sessile
underneath your
organisms
skin.
that need something on which to attach.
Different coral there.
Wow, it looks like we have a
Would you like me to
a
follow
lot
you
more
around?
rock
You're
here,
off in my
and
port.
this looks like another
I'll
black coral
scan
species
this and then
to
come
me.
back. You don't
Copy
have to
that.
worry about it.
Just slight tilt up so we can see the whole thing as you come up.
Go ahead.
See another small fish at the base.
Do you want to look at this coral, Watchley? Let's take a
Yes, Pilot, let's take a look at it, please. Thanks.
There's actually a little medusa, or bentha medusa or something like down there, it looks like, maybe. Or no, it's a squat lobster. High hopes.
It's a fish.
See some interesting organisms here that we haven't seen on this dive yet. It does look like a squat lobster living within the polyps.
Yeah, that might be a Europtychus squat lobster.
And what is the coral we're looking at? Is this another black coral?
It's another black coral, and if I don't know if anybody online knows the. Oh, there's two squat lobsters on this.
Oh yeah.
So this genus Paranthropathes is the black coral, and it's quite common for this genus of squat lobsters to be an associate on black corals, on chrysogorgian corals. Ah, that's beautiful.
Now you can see why they're called black corals. The primary stem does have a black nature to it.
Yeah, the skeleton is black on black corals.
And this genus of squat lobsters is found with this particular species of black corals. Often they're associates that often, or almost all the time, occur together. So that's one of the mysteries, is how do the young squat lobsters find their coral where they will live for a long time.
Might be too late. There's a isopod or an amphipod. I can't see. A little pinkish mobile organism there.
Let me get a little closer for you, Ron, before we get too much zoom up.
Yep.
And Cheryl, what's the undulating? Is that a sponge of some type?
I don't know. It actually
Quite
looks more like
guy there.
some kind of egg case or something.
You get lasers on that guy.
Lasers coming on.
Any ideas from the chat room of what this sack-like undulating
Lasers are on.
mesh is?
Want to sweep it to the bug, or?
Yeah, we want to sweep into the bug first. The bug.
It looks like a little amphipod to me. We saw something like this previously. A little pink amphipod.
Yeah, we didn't get a good look at it before, so this is maybe better view.
Trying to hold it for you.
I wonder what that.
He's small. I
green,
might have to put it
I guess that's another sponge.
Up.
Right below the amphipod.
Back zoom.
Thanks.
Yeah, those are little green sponges. You can see one at the top of the screen.
Oh yeah, look at on the left. Yeah, I see.
And I don't know if that's what he's crawling on is either a hydroid or a very small black coral.
It's a brittle star's tentacles coming out on the bottom there. Must be his home.
This rock looks like it has a lot of cavities for things to hide in.
It's a little microcosm.
We're at 1,320 meters here. I'd say there's at least 3 or 4 different sponge species that you can see in this view here.
Okay, Roland, when you're ready.
Video is clear.
I'll go down a little undulation here, see if we can get a better idea what that is.
You can see the brittle star arms that are coming out of the hole right below the amphipod.
Hermit crab on the right of your screen, just moving off screen.
Looks like that little fish hid. Looks like the little fish who's at the bottom of the rock is hidden himself in HD2. Don't see him anymore.
When you get it.
Yeah. Okay. go over a couple other these little rocky parts and come around back on track.
Do you want me to swing over a couple of degrees and begin to lose your light
So
pool?
this is the type of landscape we were expecting to find on this dive, so we're very happy we were so close to it. The map did not lie.
That's an amazing thing to have the great mapping that we have now, so that we can be very accurate where we put the ROV and have a very educated guess of what might be here.
Yeah. And you can see how many of these rocks are tabular in form, and they're broken blocks. So these were part
Floaty
of
top center.
sedimentary layers for certain, and they're all stacked up underneath the sediment. And these are the edges of those layers. That's a beautiful view.
It's getting close. Up into our light pool.
This is larvation. Actually, this is a really nice view of when you can see the very, very transparent outer filter house has some specks on it. But what you can see really well here is the very complicated, intricate inner filter with different lobes.
Nice
and
fire.
that's actually the one that collects particles that they're feeding on. The outer house is actually excluding particles that are too large for what they actually want to consume.
That's great. Thank you, Stephanie. That is the voice of our sampling lead, who is busy right now trying to log those samples we took earlier. But her name is Stephanie Bush, and she is from the Smithsonian.
You might tilt up.
And it looks like we're seeing more Stichopathes black corals, and this is the orange variety that I mentioned before that I've seen more frequently than the yellow, although Scott France said that yellow is actually a very common color for Stichopathes as well.
There's a pencil urchin to its right on the rock next to it.
Pencil urchins are in the genus Sideris, or at least some of them are. I'm not sure if this one is Sideris genus or another. But these spines are a little bit more thin than the Sideris that I'm used to, so this might be a different genus.
Lasers off.
We have a stalk of a former coral to the left of the urchin.
Keep rotating left. There's something interesting on the left edge.
Yeah, it looks like there's a Bersingid starfish to the bottom left.
The rock
maybe
pattern on it.
Oh, I see.
Yeah, that's interesting.
Is that a collectible rock?
That looks like it might be a
fossil.
Fossil, yeah, maybe a flabellum coral, which grows on, well, actually can live on soft sediment. Okay, and there's a close-up of the Stichopathes.
Is that a solitary coral, the fossilized one?
Yep, solitary coral.
Sorry. I might get a ship move a little closer to us here. Yeah. Yeah, you see he's got some sonar returns out here. Maybe come out this way.
Video's clear.
Yeah.
It's a little squat lobster on the rock on the left side of your view.
I could agree with that. About 20 meters towards that.
If I were to be a rock collector, I might want that one in HD2 next to your right skid.
Yeah, ask if we could, since we're kind of stopped here, we could get our rock sample for representing this area.
Yeah, let's come wide. I'll back up a little bit. We can look around and see what our options are.
Like plenty of.
There is a piece down here, at the very bottom of the screen right now. That's a good size, or an okay size. There's a couple pieces up top that could work.
And if anything you can find has an organism on it,
2, 4,
that'd be a
5,
bonus.
20 meters.
Okay. Just so you know,
Can I get another ship move, please?
having an organism.
20 meters.
That's true in the rock box. Probably wouldn't survive transport.
Yeah, it might not survive, so it would limit our size.
Yeah.
To the bio box versus the rock box.
Orange anemone on the
but
right side.
if there's one that you see that jumps out, there's a little squarish blocky one just to the right of this large one.
And a big
looks like a good shape. I don't know if that's a good one.
Right in the middle of the screen.
Or the background.
So the huge one in the middle, but just off to its right, like kind of tucked under.
Oh, yes. Okay.
That's kind of that's a good size. It also doesn't look like it's just crust, maybe.
Okay.
Hard to tell. I don't know. If you have any.
No, that works for me.
I can poke it and see if it's loose.
The bigger one would be great for the large rock box, and then we'd have our major collection for the day taken care of. And maybe some of those encrusting organisms will survive. It's a good thing this is a little shallower here. We have a longer dive than we've had the last 2 days because there's so much to see. This is great.
Spinning up hydraulics. Hydraulics are spun up, ready to go when you are.
Ready?
Ready to go.
Here comes the arm.
And there's another, I think, a Stauropathes black coral to the left of this rock that we're trying to sample. This will be the first geology sample of our dive.
Oh, that time it worked. Just watch this and tell me if I'm going insane. It's probably going to go great from here.
And on the ROV, we have some special boxes just for rocks. We have 2 special boxes, and
would you want to open
it's
up the
very
rock
helpful
box
to
first?
keep the rocks in a different place than the biology specimens, because I think
I guess this isn't as large as I thought it was.
The biology wouldn't fare so well.
Yeah, let me prod it real quick. Yeah, it's definitely loose.
Is the larger of the 2 possible?
This one here?
Yeah, or is that just too large?
Let's get the lasers on. I have a feeling it's too large, but we can check for you.
Lasers coming on.
Lasers are on?
I believe so.
Bank 1. There. I'm going to have to say definitely not going to fit in our rock box.
That looks pretty big.
I'll take what I can get. The smaller one looks great.
Okay.
Thanks.
I was picking the smaller one on its shape. I don't know if there's something better around that you saw.
No, don't worry. That's
okay.
All of this should be the same material. So once it gets cleaned off of the sediment, it should be a great specimen. Be nice to have a fresh exposed surface of it, but we may not have that until it's back in the lab. It's pretty hard to identify what it is. It certainly has, it's sedimentary. We know that for sure. It is not a lava. If you were following along with the Pacific dives, we're not seeing lava here. This is a sedimentary rock. Black material is probably an iron manganese crust. May even have some phosphate in it, phosphorite, which we see northwest of here at the Charleston Bump. We see a fair amount of manganese phosphorite deposits, and they're very solid. And these are minerals that precipitate out of the seawater. So imagine if you had, for example, a glass of salt water and you let it evaporate, you'd have salt crystals. That's what we mean by precipitating from the seawater. In this case, it's not from evaporation, it's from too many of those ions of manganese and phosphate, if there is phosphate here, but also iron. So the seawater has a lot of dissolved